at the very beginning, I want to provide some domain background so that you can understand what's the difference between RNA and protein. And then I want to talk about more about what are the challenges in RNA structure prediction, and if we have AlphaFold for proteins, why can't we actually make it so easy to work for RNAs? Then I will go to our approach, so what I've done, and I will present some results and summary. All right, so let's start with RNA versus protein. The fundamental difference is the building blocks of RNA and the proteins. So in RNA, we've got only 4 building blocks, and we call them nucleotides. And we've got cytosine, guanine, uracil, and adenine. What we can say about that is that they usually create something like canonical pairing. So usually what we observe in nature is that cytosine is paired with guanine, and uracil is paired with adenine. In contrast, in proteins, we've got 20 amino acids, so the amount of building blocks is much higher. What is also important about RNA is that the blocks, they are very, let's say, flexible. So usually it's called canonical base pairs. So usually cytosine is paired with guanine or with uracil in that way. But it happens quite often that we can change it in any way. Like, for example, guanine can pair with uracil, or cytosine can pair with adenine, or something can flip upside down. It still creates a valid base pair. And then we call it like non-canonical pairing. Another key difference is the number of degrees of freedom. So proteins in general are very, very rigid. So in proteins, we've got, what you can see on the right, only 2 torsion angles to. So the torsion angle says how the building blocks are rotated in the context of each other. And in RNAs, we've got 7 torsion angles that precisely describe the structure. And even if we skip at least one of them, the description is not sufficient, and we are not able to reconstruct the structure by using fewer amount of torsion angles. So you may ask yourself, if we can do it, if AlphaFold 2 was able to solve the protein structure prediction, why people can't just use the same approach to predict RNAs? And in the next couple of slides, I will explore that, and I will explain why it is actually a problem. The most important thing that you should remember from this slide is how AlphaFold 2 works and what are the crucial parts of AlphaFold 2. So the very important parts are multiple sequence alignment, which is called here MSA, and templates. So in principle, it means that once you have a sequence, you can align that sequence with many other sequences from databases. You can search for template structures. So you can, for example, choose that this fragment is very similar to what we observe in this particular structure. So I can use this fragment and try to adapt it to my structure, to my prediction. But in RNAs, it doesn't always work in that way. And finally, why is it important? I would just say that this year's Nobel Prizes were in actually the guys on the left that you can see on the left. Those guys were awarded for discovery of microRNA and its function, and the guys on the right they were awarded for computational protein design and protein structure prediction. And what we are trying to do here in my work is RNA is like combining both, so it's RNA structure prediction. So from biological point of view, this problem is valid and it's important. So now I would like to talk about the challenges in RNA 3D structure prediction. And the first one is the data. The first one is the amount of data. So what you can see here, this chart presents the amount of PDB structures in PDB database. And what you can see here is that for proteins, we've got like over 200,000 experimentally determined structures. In contrast, for RNA, we've got only 8,000. And what is even more important, that a lot of them is actually redundant. So it means that we have multiple structures. They come from different organisms, but actually they are made up from the same sequence of the structures identical. So if we remove the redundancy, it will drop to even below 2,000 structures. Another problem is high data imbalance. So again, what you can see here on the x-axis is the number of entries, and sorry, on the y-axis you can see the number of entries, and on the x you can see the RNA families. So RNA family is a group of structures that have some specific function. And what you can see, there are two bars like rRNA for rRNA and tRNA. And over 80% of our database of our structures belongs to only two protein families. And this makes our data set highly imbalanced. And this is kind of common problem that many methods is just biased to only predict structures from those two domains, but fails to predict other. There are also other challenges. So the first two RNA content in protein data bank and data imbalance. There are two that I mentioned in two previous slides, but it's not all of them. We also have others. So determining high quality structure for RNA is a bit more complicated than in proteins. So it often happens that we have problems with quality of experimental data. And another problem is the sequences and sequence alignment. So as I mentioned in AlphaFold slide, multiple sequence alignment is kind of very important strategy in structure prediction. And for RNAs, there are two problems. Like, first of all, it's a bit more complicated to create a reliable alignment for RNA. And the second problem is, again, the lack of data. So if you have low amount of data, low amount of other sequences, it's very difficult to make a valid sequence alignment. So now I'd like to present our approach, like how we want to address those limitations and those challenges. First of all, the problem was with amount of data. So we were thinking that maybe we could try to do some, let's say, maybe kind of augmentation or just to try to extend our data set. So instead of predicting the full structures, maybe we could start with predicting smaller particles or smaller substructures. And those substructures are called the local 3D RNA descriptors. And what you can see here, how we construct those substructures, those descriptors, we just choose one specific atom in the structure and create a sphere of a certain size, like for example, 16 angstroms. And everything that comes inside those sphere is our substructure, so we can save it, we can store it as a separate PDB file, and then use for training. And we repeat that process, which you can see on the bottom. We repeat that process for the whole structure. So here is our, let's say, data workflow. So at the moment when we started working on that, we've got like 1,500 non-redundant structures. And we extracted those descriptors in a way that I presented in the previous slide. And we've got almost like 180,000 descriptors, but many of them was redundant, and this redundancy comes from the fact that, you know, like if we have smaller substructures, it quite often might happen that certain part is almost identical in many structures. So we have to, you know, like do spatial alignment and remove the redundancy. So we ended up with 44,000 substructures. And then we split that into training and test set. The important thing that you can see here is that in the training set, we've got 33,000 structures. And to kind of address the imbalance problem, we split them in a way that in training set, we use only rRNA and tRNA, so the majority classes in our training set. And in the test set, for testing, we used substructures from all the other RNA families that were left. This is the architecture of our model. So what you can see here is a couple of components. First of all, there's the user input, so user can indicate a single strand or double strands or 3 strands or as many as he or she wants. And in the strand, you can indicate a separate strand. You indicate the nucleotide sequence and also dot bracket notation. So dot bracket notation means which nucleotides should create a canonical base pair. This is something I presented at the very first slide. Then we have other components like graph neural network, RNA language model, and transformer. I will explain those components in a while, the thing I will start with is denoising diffusion model. So how we use this generative property to predict the structure. So we start with random point cloud, and we try to recover the structure. And this slide presents it in a bit different way. Like we have, what you can see here on the left is our original graph of atoms. And what we do here, we just add noise to atoms' positions. So the connections or how the atoms and residues are built, it remains unchanged. But everything that changes, like the only thing that changes is the atom's coordinates. So we add noise to atom coordinates, and we train our generative model to reverse that process to predict atoms' coordinates. Our representation of the nucleotides is coarse-grained because this is example of RNA. You can see that there's a lot of atoms to process. So when we use graph neural network, where each atom is a single node of a graph, it's kind of memory inefficient and computational inefficient. So we had to somehow simplify the problem. So that's why we use only 5 atoms in our representation. So this is called coarse-grained representation. And we use only 5 atoms in our graph. Now I would like to say a few words more about our graph neural network, how it works. So it consists of, let's say, 2 layers. Each layer is responsible for, let's say, a specific type of interactions. Like, for example, one layer is responsible for local interactions only. And in local interactions, we can measure the angle between 2 atoms, or we can measure the dihedral angle between 4 atoms, or we can just measure the distance. While in long-range distances, long-range interaction layer, we just measure the distances between the atoms. So what the neural network is learning, it's learning the distribution of the angles and distances between atoms. The second component is the RNA language, and I would like to explain why do we use that. The reason is quite simple, because RNA language models, they provide quite rich inductive bias. So they provide quite a lot of information about the structure. Although it's only the sequence, they still provide quite rich information about the structure. And this is the example of how our model performed before we used our language model. So maybe it's difficult to see, but in principle, what you can see here on the structures on the top or on the bottom is that we have canonical base pairs. And those canonical base pairs, they are like planar, so the structure keeps those canonical base pairs. But in the middle, there were some non-canonical interactions that we did not indicate. And the model tried to behave like, if we don't have any interactions, I want to push those residues as far as possible. And that's why this structure is not very good. It has quite weird shape. After adding the RNA language model, suddenly we've got really nice helical shapes. So suddenly the model has more context, and it means that, let's say, we've got still the canonical base pairs, but it doesn't necessarily mean that in places where we do not indicate any interactions, that it doesn't necessarily mean that we don't really have those interactions. Sometimes there might be non-canonical interactions, so they might be like shifted or translated somehow or rotated upside down and still create a reliable structure. Our main competitor at the time when we were working on that was AlphaFold 3. When you take a look at the slide of the AlphaFold 3 structure, you may notice that many components are exactly the same that were in AlphaFold 2. The main difference is the diffusion module at the very end. And it's not, you cannot see it on the graph, but on this chart, but the diffusion model in AlphaFold 3 actually does not use graph neural network. And they even claim in the publication that they are aware that graph neural networks are just state of the art, but for some reason, they just rely purely on transformer. We measured at our test set And here I just selected a few samples from our test set to see the performance of our method. And it seems that it's not that bad. And in general impression, when we compare our method to AlphaFold, and actually when I was dealing with AlphaFold 3 and comparing the results pure only for RNA structures, was that AlphaFold still, for RNAs, it doesn't work that well like for proteins. And we can easily observe some failures, like for example, some clashes or some strands colliding with each other, or the general tendency to produce every single structure to fold it in helical shape, although it's not always helical. Another thing that we wanted to measure is how well our method keeps the interactions between atoms, between residues. So for us, it was kind of important to keep the interactions that we indicated to the model. So when we expect the model to keep certain interactions, we don't want the model to disturb them. In AlphaFold 3, you cannot do that. Like, you cannot say what kind of interactions there should be. The model needs to, let's say, guess it on its own using multiple sequence alignment and so on and so forth. And from our perspective, when we talk to, let's say, experts in RNA structure prediction, for them, it's kind of a bug or missing feature in AlphaFold 3 that you cannot indicate the structure that you want, because in our approach, you can kind of modify the secondary structure of the interactions and try to run it again and to rebuild the structure with a bit different interactions. All right, so to sum up, I would like to say that our approach, the novel, it's kind of novel, like using RNA 3D descriptors is kind of novel approach in the RNA 3D structure prediction domain. And we show that it's able to predict reliable 3D structures. And what is important, we did not use structural templates. So we kind of omit the problem of missing data, but there's still a lot of work to be done to predict full atomic structures and how to assemble the bigger structures. Another thing is that we proved that embeddings derived from RNA language model, they have significant inductive bias and they significantly improve the performance of our model. And finally, what I wanted to say is that the secondary structure defined by the user is a very useful feature, and it's not a bug. It's just something that domain experts expect in the domain. As the end notes, I want to say that our work is like fully open source. We've got GitHub code, and we've got Zenodo database. We use Zenodo database to share the model weights and the data set, and this work was accepted to NeoRIP's workshop, Machine Learning in Structural Biology. And actually, this is it. I want to thank you, my supervisors, for all the support in this work. So yeah, thank you so much.